Hey everybody, and welcome to chapter number 4. This chapter we are talking about how we recognize objects, and just to be specific, when we say recognize objects, we're mostly talking about visual object recognition. In fact, I would say this chapter is almost exclusively visual object recognition. We'll talk about other forms of recognition in chapters 6, 7, and 8 when we talk about memory, but for now, think of this as kind of like a bridge from the last chapter, which is all about visual processing, how we are able to make sense of the visual world around us, to the next chapter, which is going to be chapter 5, which is attention. So we're kind of bridging the gap between those two. So let's get into it. As I mentioned with last chapter, if you've taken sensation perception, you're at a little bit of an advantage because you're going to see some overlapping ideas here, especially when we talk about face perception. Which is the last thing we'll be talking about here, and also recognition by components, which is a theory about object recognition that we talk about in sensation perception as well. But if you haven't taken sensation perception, don't worry about it because we're going to be covering it all. And last thing that I'm going to mention before we get into it, you may see on the PowerPoint that this slideshow has like 150 slides. We're going to get through this in an hour, roughly. Don't let the 150 slides fool you. A lot of that is going to be interactive at the very, very end, whenever I test your memory for specific things. So let's get into it. What we're going to be talking about is we're going to start by talking about visual search, which is something that we talked about in chapter 1. We're going to come back to it, talk a little bit about that, and then we are going to talk about exactly how visual search is related to our understanding of recognizing the world around us. So let's get into it. I got a lot of videos for you here. I'm gonna say, if you had to choose one of these 4, I would go with this one right here, the one that says newer prosopagnosia video. It comes from a news clip where you're going to see basically like this guy, a news anchor, kind of interview some people that have face blindness. So check that out. It's really, really interesting, but if you're really pressed for time, this older video is shorter. But it's, it's like 3 minutes or so, but you can tell that it's like from the early 90s or whatever. And I like showing you new content whenever I can, so I would go with the newer video if you have time. And then these others are just kind of, optional. But we'll get into prosopagnosia later at the very end of this chapter. So let's get into it. Chapter 4. So let's talk about visual search. Visual search is a cognitive task that you see a lot because it's been around kind of since the inception of cognitive psychology. And the premise is very, very simple. You're just asking, where is the target? Or is the target present, yes or no? That's what you have to do usually. So in this case, the target is going to be the vertical line. So I want you to find the vertical line standing up, and I want you to actually try this, actually try to be interactive, because I'm going to ask you to do this, and I want you to think about what's going on in your mind whenever you're trying to solve this. All right, so here we go. Find the vertical line. Okay, so that probably popped out at you immediately, right? So right here, we got the vertical line popping out immediately. Why does it pop out? Because it is totally different from all the others. Okay, that was easy enough. Your reaction time on that should have been very, very quick, right? So you'd probably have really low reaction times, like maybe 60 milliseconds or something like that. Really, really fast. All right, let's look at the next one. So for test number 2, I want you to find the green colored line. So here we go. 3, 2, 1. Okay, yeah, and again, this thing pops out right at you, right? Again, we're talking very, very quick, maybe 60 milliseconds or whatever that you're able to spot this. All right, so now for the final visual search, I'm going to ask you, I want you to find the vertical red colored line. So it's the one standing up that's also red. Now here, I'm doing two, and I'm kind of spoiling, well, actually no, I'm going to go ahead and show you. So I want you to try to find this, think about how long it takes you, here we go. Alright, so this one you probably found pretty quickly. It's this one right here. But I bet that it took you a little bit longer. Maybe not a lot longer, but maybe just a fraction of a second longer. So maybe this one took you 100 milliseconds instead of 60. 100 milliseconds, very, very fast. It's a tenth of a second, right? But that is almost twice as long as 60 milliseconds, which is generally what you would get for the first two. So why is this one harder? Why is this one longer? It shouldn't be, right? It shouldn't take you longer to find this one because you already showed me that you can find vertical lines and you can already find lines based on their specific color, right? You did that already in the first two. So why is it that whenever you have to do both together that it seems to be a little bit longer? The reason why is because this target shares features with the distractors. So this one is up and down, which is fine, but there are also others that are up and down. They just happen to be green, right? So they are not the ones we're looking for. But we're also looking for red, but again, this isn't the only line that's red. Red is a shared feature with these other distractors. So whenever we are trying to find the red line that is up and down, we have to kind of satisfy these two characteristics, that it is both up and down and that it is both red. That is very different than this task right here, where essentially you're just kind of looking for the one trait that isn't present in the others, right? It's unique. It pops out at you because it is green, or it pops out at you because it is vertical. So what does this mean? Basically, it means that whenever we are trying to search for objects around us, the target is important, but it may not be as important as the surrounding environment, that these distractors, these other irrelevant stimuli, how much they share in common with your target makes this more difficult or less difficult. So why is that? The reason why it was harder is because we were looking for a combination of features here. So the lesson is that the more features the target has in common with its distractors, the harder it is to find. And there's a good chance you already knew this because you've probably seen this game before, right? If you haven't seen this, well, you've seen this before, right? Probably most of you have seen this before. It's Where's Waldo? This was all the rage whenever I was growing up. You get these books from the book fair in elementary school, and you got to try to find Waldo. He's this kind of weird, funny-looking guy right here, and you can find him because he is. I'm going to let you, if you want to pause it and try to find him here, go for it. But he is right here. There he is. So why is Waldo hard to find in these games? Is not because he is common or basic or, like unremarkable. He's a funny looking dude, right? He's super tall and he wears this very ostentatious red and white hoodie and he's got this red and white beanie. It'd be hard for him to stand out even more than what he is, right? If you were to come in the classroom wearing something like that, I'd probably assume that you wanted attention, right? Because red and white stripes like that, you're trying to make a statement. So why is it that Waldo is oftentimes hard to find here? The reason why is because the environments around him share a lot of features. We see lots and lots and lots of white and red stripes all over. And these are meant to kind of grab our attention, right? Because they share features. They share a lot of the same features as our target. So that makes this search hard. If I went through and basically gave you this exact same tableau, but instead, like everything was black and white except for Waldo, he would pop out at you immediately. And there, it's not the case that, we're distracted by how much is around him, because we still have a lot of things around him that are just in black and white. The difference is he's now unique and he pops out because he's not sharing features with his distracting environment. All right, so to change gears a little bit, let's talk a little bit about how, or our understanding of how we are able to detect Waldo to begin with. The book talks about feature net models. Feature net models are a little bit frustrating to deal with because it's important. I want to make sure that we're all on the same page here, that when the book talks about a feature net model, this is a model. In other words, it is a representation. It is a metaphor for how this works in the brain. This, what you see here, this image, is a simplification of what's actually happening. But again, it's a metaphor for what's happening. It symbolizes what happened. It represents what's happening. That's what makes it a model. So let's say that you're trying to find the word clock. Or not that you're trying to find the word clock. You're looking at this word, and you will eventually, because you can read, understand that this word is clock. How are you able to do that? How are you able to read, right? So what the feature net model supposes is that the earliest neurons in our perceptual system are going to be gathering this information right here. They're going to be gathering information on the most basic features that we can see. That means lines and edges and curves, right? Basic features. So here we'd have this neuron right here that normally detects this kind of curve like that. It is really active. We see this C, and so that neuron in your occipital lobe, that ganglion cell, is going to be firing like crazy because it is sensitive to that specific curve. So we know that specific curve is active. We also know that the similar cells that are activated whenever they see this and when they see this down here, they are also active. And so we are going to have other networks that are going to be detecting letters, and those are going to say, hey, we got all of the right features for us to be able to put together the letter C. So we recognize the letter C. And we're going to do the same thing for the letter L. In this part of our visual field, we're going to have some cells that are detecting straight up and down, and some cells that are going to be detecting back and forth, left and right. And because we detect both of those cells to be active, we recognize that as the letter L. We're going to do the same thing for the letter O, the same thing for the letter C, and the same thing for the letter K. Whenever we recognize we got a C, an L, an O, a C, and a K, then there are going to be other networks that are going to recognize the pattern of those letters so that we recognize it as the word clock. So, in other words, whenever we are trying to recognize words, the steps of this process is that we're first going to recognize the features, then we're going to combine those features so that we recognize them as letters, and then we're going to combine those letters so that we recognize them as words. Easier said than done, but that is essentially the flow of information in the brain. So we talked about top-down regulation in Chapter 3. The book talks about it in a little bit more detail here in Chapter 4. And they do so by talking about how your understanding of the world around you can affect what you perceive. So, for example, the word corn. We know what the word corn is, we know what it represents, we know that you can eat corn, etc., right? So corn is not a mystery to us. But if you'd never seen this word before, if you were speaking a totally different language, you might not know if this is a word or not, right? And so why is that important? Because if we are going down and we are showing someone CQRN, like that kind of looks like the word corn. But we know it's not, right? We know that that isn't the word corn. We know that's probably not a real word. So why is that? Well, we start out with the word itself, CQRN. Then, just like we saw in the last slide, we're going to be detecting the individual features of these letters. When we establish the individual features, then we're going to combine them so that we recognize the letters. And then when we recognize the letters, we're going to be recognizing the bigram detectors. Now, the bigram detectors are, according to the book, kind of this intermediary stage. So they help us kind of put together little bits, little pairings of these letters. And then from the little pairings of these letters, we recognize the whole. This is why if you were to very quickly be flashed this word, CQRN, you'd probably recognize it as CORN. Why? Because of top-down regulation, because of top-down processing. Why is that? Because of just the basic probabilities that you understand about the English language. You know that CQRN is a very unlikely combination of letters. You know that CORN is a very common pattern of letters because it spells a word. A very common word, actually. So, top-down processing means that whenever we see this right here, CQRN, we might be likely to recognize it as CORN because it shares so many of the same features and is more likely to have occurred. So the probability helps us get there. Similar to top-down in the same, continuing the same train of thought, this is why whenever we talked in Chapter 3 about these examples, we generally recognize this as THE, and we recognize this as CAT. And the reason why is because we know the basic probabilities of language, that TAE is not a very common word, nor is it a very common, combination of letters. CAT is, and THE also is. Those are more frequent, they're more probable, and so we perceive them first. That is an example of the context influencing your perception, or of top-down processing affecting your perception. Okay, so we're at the 15-minute mark. Now's a good time to take a quick break if you're feeling a need to do that. We're going to kind of move into talking about how we are able to use feature binding to combine different features and different combination of features together. All right, so this picks up again from chapter 3 when we were talking about illusory conjunctions or conjunction errors. This notion that whenever we are not paying appropriate attention, that our visual world is very fragmented and it's not quite bound together because of parallel processing. We have the location of an object and its identity processed in two separate streams of information, parallel processing, and if we're not paying attention, then those separate streams of information may confuse one another. We may not have a solid grasp of the visual information in front of us unless we're paying attention. So let me give you an example of that. This Treisman and Gilad experiment, this is from over 30 years ago. Wow, over 40 years ago. And it is a fantastic paper. I love this paper. I would force you to read it, but it's a little long and it's got like 12 separate experiments in it. And I know that students generally don't like that. But Anne Treisman, she's going to come up, I'm going to say at least 2 more times after this chapter. Anne Treisman is super underrated. She is one of the best experimental psychologists, in my opinion, of the 1900s. And she's no longer with us. She passed away in the mid-2010s, I believe. I still remember when I was teaching cognitive psychology the semester that she passed away. She's one of my favorite psychologists, so you're going to hear about her a couple of times this semester. But this idea about the binding problem, she essentially provided the explanation that most people adhere to these days. So if you remember the binding problem, this idea about how is it that we're able to combine these features in our mind whenever they are totally separate from one another in separate parts of the brain, she was able to kind of show how we're able to do that using behavioral experiments. So she was interested in how these features are bound. Here is how she did it. I'm going to give you a very quick kind of demonstration of one of the tasks that she used. But she was really interested in this idea of illusory conjunctions, and she was very interested in how if we're not paying close attention, we can have illusory conjunctions or illusory conjunction errors take place in front of us. If I had to give you a real-world example of conjunction error, they don't happen all that much, or at least they happen all the time. It's just that we're not really aware of them all that much. But whenever I am aware of them, it's almost always because of something that I've read, or I'm looking at a page or a website, and I see 2 words that are kind of close together, and I mistake them for being a separate word entirely. So, for example, if I was reading a book and I saw the word Western Field, I might initially confuse that with the word Westfield. But whenever I look at it more closely and pay attention to it, then I can see it doesn't say Western Field at all, it says Westfield. That's just a very kind of easy example of how you would see this in the real world. Alright, so to give you a taste for what Anne Treisman and her collaborator Gilad did in 1980, I want you to focus on this fixation cue. I don't think we've talked about fixation cues this semester yet, but they're going to come up in almost every single cognitive task that exists. A fixation cue is usually a little cross, and it's usually here in the middle of the screen, and it is meant to be where your participants are looking. This is where you want them to rest their eyes. This is where you want their eyes to start out. Why is that important? Because a lot of times we're really interested in reaction time and response time, how quickly people can respond to a task, and so we want to make sure that everybody's basically looking in the same area. Because if you're doing something like a visual search task, which is what we did at the very beginning of this chapter, if somebody always starts in the top left corner and then moves over to the right, and then they come back and they move over to, that's how they are searching from it, fine. But what if somebody starts here in the center and then, starts spiraling out as they're searching for their target? The 2 different kinds of ways that people might tackle that are going to change their response times. So we generally want people to kind of start in the exact same place, which is going to be in the center of the screen. So that's why you see this cross in almost every single experiment that we're going to be doing this semester. All right, so you get your eyes fixated here. And so in a moment, I'm going to ask you about what you see on the screen. Here we go. Here we go. Okay, all right, so good chance that you saw this object right here, right? There was an object that appeared right here, and it was blue, and it was a square. But you probably saw something over here too. You may have seen something in the top right-hand corner of the screen. I'm not going to play it back for you, because now that you know where it is, you can put your attention over here, and you're going to see it very clearly, and you're not going to have an illusory conjunction. This is, by the way, also one of the disadvantages of having this online, is that this is one of those things that I like to do in the classroom because I get so many different responses. Or I ask the class, what did you see right here? And I'm going to get a lot of different responses. Some people will say, I saw something green, or I saw something blue and yellow. And then I say, okay, what did you see that was blue and yellow? I don't know, I saw a square and a circle, or whatever. Here's what you saw. Yellow and red, right? So a yellow triangle and a red square. But there's a good chance that you may not have seen both of these, or you may have only seen one object, or you confuse them. Either way, if you make any kind of error where you say that you thought that the yellow, that the square was yellow and that the triangle was red, or that you mismatched their colors, or the shapes or whatever, that is an example of an illusory conjunction. Sorry, I have a gnat in this room for some reason. So that is an illusory conjunction. Now, if I were to backtrack and then ask you the same thing, again, you're supposed to be looking right here, but you know that it's going to pop up over here, I'm going to play it in just the same amount of time, but because you know where to put your attention, you'll be able to see it. All right, right? So whenever you saw it, you saw it. Okay, so for trial number 2, now that you have a general idea for how this works, let's give it a shot. All right, so you're looking here in the center. I'm going to show something very quickly. Okay, so what about that one? You saw a blue rectangle here, but what did you see over here? Again, if I were to ask you on these, you'd probably be right that you saw a blue and a black object, but you may not know which one was on the left, which one was on the right, which one was on the top, which one was on the bottom, which, what their shapes were, things like that. So I'm going to backtrack again. Actually, close your eyes. Alright, so if we're going to do this again, I'm going to try to do this in the same speed, but now you know where it is. So I want you to look down in that corner and let me know if you can tell me what you see. Okay, whenever your eyes are down there, it's very, very clear what it is. It's a black arrow and a blue heart. Yeah. Alright, let's do one more trial. So again, you're looking right here. Here we go. Okay. So in this case, you saw something distract you, this big red thing up here. But what did you see right here? This one is usually a little bit easier, and the reason why it's a little bit easier is because it is closer to the center of our visual field and closer to the object that was distracting us. And so we have some of our attention spread out over here, so you may have noticed that there was a purple moon and a yellow cross. And then finally, here we go, trial number 4. Okay, so you saw the lightning bolt here, but what did you see over here? Close your eyes, I'll take you through it one more time. Here we go. Alright, here we go. 3, 2, 1. Okay, if you're looking right at it, then you may have seen the word newt and the word purse. But what happens is that if you do this for a short enough time, people will think that they see the word nurse. Why is that? Because they are making an illusory conjunction. They are combining these features together to give you an error. So illusory conjunctions are whenever 2 or more things appear close to each other in the visual field and they are crowded, and for that reason we're not paying attention to it. Those features are not solidified. And we have sometimes we will have where they combine incorrectly, which is why you may have seen a blue arrow and a black heart instead of a black arrow and a blue heart. Alright, so that is feature binding and the binding problem. All right, so let's get into how we can take many features and make them match an object. Here is another paper that I was going to recommend that you read, but this one is actually even longer than the Treisman and Gilad article, and it actually has more experiments than that one too. This one has so many experiments in it. I want to say maybe 14 separate experiments, and you can see here it's, what, 32 pages long or something like that. It's a very long article. Let's see. Irving Biederman is he was a very famous psychologist, or a very famous researcher. He had a reputation in the field, and I'm not going to say too much more about that because he has passed away, and I shouldn't say bad things about people who have passed away. Yeah, that's all. I'm going to leave it at that. Irving Biederman came up with the recognition by components model. In your textbook, you'll see this referred to as the RBC model. The recognition by components model came out and basically got a lot of attention and praise right away. And then with neuroimaging, people have only kind of further solidified this theory. Here's one thing that's a little bit annoying about, I'm going to say psychology, in general, but specifically the classes that I teach, sensational perception and cognitive psychology. It bugs me to no end that we are talking about theories. Because whenever I was in college and I saw this, I was like, wait a second, why is this a theory still? This was 1987. This was before I was born. Like, shouldn't we know for sure now? Shouldn't we, be looking at laws instead of theories, and hypotheses? But here's the issue. A lot of things that we're talking about this semester, or in sensation perception, or in behavioral neuroscience, the reason why they are a theory is because we are pretty sure this is exactly how it works. However, it would be unethical for us to really be able to figure this out. So why would it be unethical for us to be able to really figure out if this is how, the human brain works? Because we would need to go into a human who is alive, and we would need to lesion their brain in specific areas to make sure that we could validate this theory. We can't do that. It'd be illegal, it'd be unethical, it'd be immoral for me to, be like, hey, do you want 3 points of extra credit? Cool. Meet me in this back room, and I'm going to take out parts of your brain and see how, you perceive the world. Sorry, my dog visited us, and so I'm gonna show him to you. Usually I like to wait till the end to show you. I do not like when he licks my face. Alright, so that is Hopper. He was trying to get underneath the desk because I think he likes little caves or little burrows or whatever. But anyways, so that is why we can't really, why this is still a theory, is because it's just one of the things that if we were to test it, it would require us to tear apart a living person's brain, essentially. There may be a time where neuroimaging can help kind of make this more concrete, but hey, I'll let you know when that happens. Alright, let's get into it. So recognition by components, there are 3 stages in this theory, and if you took sensation perception, this should be very familiar to you. So the 3 stages of visual object recognition are, the first, we're going to take a complex object and we are going to break it down into smaller objects, these sub-objects. These are going to be called geons. What the heck is a geon? A geon is a geometric ion. And for some reason, this is one of those things that a lot of students remember from the class. Like, I'll have people 2 years, after they walk across the stage for graduation, they're like, hey, I remember cognitive psychology. Hey, geons, right? I'm like, yeah, I guess geons. And I think it's because it sounds fun to say and it's easy to remember that it's a geometric ion, but a geon is a smaller part of a larger object. So, for this lampshade, for example, one geon is going to be this cylinder and the other is going to be this kind of funnel-looking shape. For a flashlight, we got 3 geons. We got this cylinder right here, and then this rectangle. For this briefcase, we got 1 rectangle, and then we got like this 1 half loop. for this mug, we got a cylinder, and then we got this half loop. For this pail, we have a cylinder and a half loop. You get the idea. All right. If we look at even more complex figures, such as these living organisms, we can see that like a human is just a bunch of different geons, right? That we have our arms, which are these kind of cylinders that protrude from our body, and our body is itself kind of cylindrical as well. Our legs are cylindrical. Our head is also cylindrical. We got a lot of cylinders on our body, except for my cousin Tommy, who his head is shaped like a brick. So it's not cylindrical at all. And then the same thing is true for horses or ostriches, that we could break out these really complex objects into their smaller forms. So that is the first piece here, that we're going to be breaking up a complex object into its geons. The second step then is going to be to take those sub-objects and to classify or categorize them into geons that we recognize. So if I am looking at, let me backtrack for a second. If I'm looking at these different complex objects, I broke down, them into these smaller objects, but then I named cylinder, funnel, cylinder, rectangle. That, me naming and identifying those geons, that is step number 2. So step number 2 is that we recognize exactly what geons we're dealing with. And then finally we have, and I should say there are, this number kind of varies depending on who you ask, but there's essentially 36 different kinds of geons. My professor when I was in college referred to these as kind of elemental, and what he meant by that was that, like, the periodic table of elements, basically they represent, like, for any piece of matter, if you shave enough of it off, then what you're left with is just going to be these pure pieces that can't be reduced any further from what their molecular structures are. So like you have neon and hydrogen and oxygen or whatever, and you can't reduce oxygen into smaller things unless you're kind of talking about the subatomic level. Geons are the same thing. The geons, you can take a shape, and if you can't reduce that shape any further, that makes it a geon. So that is step number 2. Step number 3 is that we are going to essentially evaluate these geons together and recognize what pattern they are in relationship to one another. So I'm going to backtrack one more time. So for example, this human and this ape, they have the same geons, right? Their geons are exactly the same. The only difference is the relationship that they have to one another. What do I mean by that? I mean that here we see that these cylinders are longer. These cylinders are shorter than the torso. Here, for the ape, that these cylinders are longer than the torso. And that's part of how we are able to establish, oh, this is a human and that this is an ape. Same thing goes for a giraffe. That a horse and a giraffe, they have identical geons. The only difference, though, is the relationship of those geons. Particularly in the neck, right? Like that this cylinder is so much longer than the others, so we know it's a giraffe because giraffe is the only type of animal that comes to mind when we talk about having these geons in having this relationship with one another. All right. Okay, so here's what Biederman did. So in that study that I was telling you about, here's what he did. He was interested in how accurate could people be and how fast people could be. So he was interested in both accuracy and response time or reaction time. All right, so if I were to ask you, what is this? Quick, as fast as you can. What is this? This is an airplane. Okay. What is this? That is an ironing board. What is this? That is a camera. What is this? That is a desk or a table. Okay. So what Biederman did is he had these people come into his lab, he'd give them a fixation cue, and then he'd give them a bunch of these objects and ask them to respond as quickly as possible what it was they were. So he was asking for them to identify. And you can even see here, this is figure number 22. Think about how complicated an article must be if it has at least 22 figures in it. That is unbelievable. All right, so he had 3 conditions. He was interested in having people respond as accurately and as fast as possible what they saw. But he had 3 conditions. The first is kind of a control condition. It's this complete condition. So you see the unedited figures, these unedited pictures. Then he also had a component deletion category, and this is where we have some geons are completely missing. So you take this ironing board, you remove this geon, and this geon, and there you go. You got the component deletion version of an ironing board. You do the same thing with the plane. You take out this piece, you take out the tail wings, you take out the jet engines, and you get this, and that's just removing those geons. We also have, for mid-segment deletion, here we are going to have some lines, some parts of lines are removed, but generally the geons are still intact. So you can see for mid-segment deletion, we got a lot of these lines removed, but that the overall the geons are intact. Kind of funny over here, you can see that there's like a letter A over here on this tail. Where that comes from is for the PDF. The PDF that I got this figure from, PDFs, it's like you can set it so that it tries to recognize text, so that you can select text in it. And I did that, and it recognized this tail as the letter A for some reason. All right, so his hypothesis was essentially that if object recognition does rely so much on the recognition of geons, then accuracy and reaction time should be at its worst whenever the geons are removed. And that is, in fact, what we see here. So here, I want you to just focus on at these longer durations, whenever you actually give people, a fifth of a second to actually look at what they're, at the image. That here, this should be reaction time. It's really kind of garbled here, but this is reaction time. So the higher, the longer it takes to respond. The lower, the shorter it takes to respond. And what we see here is that whenever you remove the components, that generally reaction time does not change across, across how long you show it to them. That is, that for the most part, reaction time is going to be worse whenever you remove geons, except for the 65 millisecond condition. There, you show somebody this for a 20th of a second, and it turns out they're really bad at doing that. But that's besides the point. The whole point here is that for reaction time, component deletion had the slowest reaction scores. All right. Here we're looking at accuracy now, and this is mean percent error, and this always annoyed me when I would see this in graduate school, because mean percent error is just the inverse of accuracy. What does that mean? Basically, it means that down here, if you scored down here, that means you did 100%. It means you had no error whatsoever, which is good. So the lower the score is, the better. The higher the score is, the worse. So if you got a 40, that means that you missed it 40% of the time. So basically, the higher the score, the worse people are. And again, you see that at this really fast exposure time, whenever you start removing the lines, that's causing the worst accuracy. But whenever you're giving people enough of a chance to look at it, what you're seeing is that whenever you remove the geons, people's accuracy are the worst there. So people's accuracy being the worst when you remove the geons. All right. Other pieces, and that was, that was a very quick, kind of short, brief summary of his findings. If you're interested, you can find the 32-page document elsewhere online. So there's more evidence for recognition by components in neurological data, as I mentioned. Visual agnosia, which is where if you damage your inferotemporal cortex, you're unable to recognize common objects. This is evidence for recognition by components because your eyes and the receptors that detect those features are all perfectly fine, which suggests that what's happening is you can still recognize lines, you can still recognize edges, but you can't actually put them together. You can't create geons out of them. And while we're talking about visual agnosia, there's actually two kinds of visual agnosia. I'm not going to test you too much on those because I think it's just more important to know what visual agnosia is overall. But the pseudomorphic kinds, we have apperceptive agnosia, which is the inability to recognize simple shapes. But we also have associative agnosia, which is the ability to recognize simple shapes but not complex shapes. So why am I bringing this up now when we're talking about recognition by components? Because what we're saying is that people that have associative agnosia, they can still recognize geons, but they can't recognize higher order, more complex shapes that are a combination of geons. So there does exist evidence of this in the brain, because we see that there are folks that have the ability to recognize a simple shape, a geon, but not combinations of them. All right. So now is a really good chance for you to pause, take a break at the 40-minute mark here, because we are entering into our final stretch, and I'm going to take a sip of this water. So why don't you join me? Get a beverage of your choice. Make sure it's non-alcoholic. Well, I don't know. You can probably drink something alcoholic here because you're not on school ground. And just don't tell me if you are. Just like I won't tell you what's actually in this. This is straight up vodka. It's actually not, just in case the president of the university is watching. I know she subscribes to this YouTube channel. This is just water. All right, let's get into it. So let's talk about face perception. Face perception also takes up a pretty good chunk of the chapter in the book. And we're going to be spending a lot of time talking about it because this is kind of a really cool, and this is going to feed into your research article number 1, which is also about face processing, which is why I wanted to spend a little bit more time talking about it here. So this is an example you can find, I think in your textbook, but also online, some early research looking at face recognition. This is from the 80s. How do you know it's from the 80s? Because this is Margaret Thatcher, who was the British Prime Minister in the 80s. So, do these faces look different? And the reason why I'm kind of laughing at this is because you can tell this is not a good Photoshop job. This was done whenever people were manipulating photographs using, Xerox scanners and stuff like that. So these faces do look different. If I were to ask you which face is the actual real face, you'd probably go ahead and tell me it's this one, right? Because you can see some lines that have been edited over on this one. But you can see that very clearly here. Whenever I turn these faces right side up, this one becomes a horror show, right? This one now looks very scary. Whereas back here, I don't think it looks so bad, right? This one looks kind of fine. You can tell it's been doctored, but it doesn't look like, a horror movie like it does over here. Why is that? That is because of something called the inversion effect. The inversion effect, the textbook talks about it. Basically, the idea is that faces particularly are hard to recognize any kind of edits or changes to them when they have been turned upside down. Here is an example of this. This is the guy who wrote the textbook that I use in sensation perception. Which one is his actual face? I will let you look at these images, and I want you to tell me which one is his actual face. Because when we turn it, now you can very easily see that this is the wrong face, right? No one's face actually looks like this. So why is it that when you look at it here, why does his face look pretty normal here? Why is that? That is the subject of the final, let's say, 20 minutes of this chapter. Okay, so if you compare that with this, letters and words and letters and words, and you look at these, you can pretty easily tell that there's something wrong with this one, right? You can tell that this letter is screwed up. And that is because, yeah, there it is, the offending R. And that is because words and letters are not impacted by the inversion effect the same way that faces are. Again, we can see taxi. If I were to ask you which one of these is correct, you would know this one is correct, and then this one is upside down. So again, not hard to do whenever you're dealing with things that are not faces. So faces that are inverted are especially difficult to detect, and the reason why is because, and this is kind of a, sums up the next 20 slides, basically, is that the reason why is because faces are not processed in the brain the same way as common objects are. We perceive them differently than other objects. But you already knew this, because at the very beginning of this chapter I was talking about prosopagnosia, which is an example of whenever somebody damages their fusiform face area, or the fusiform gyrus, sometimes people call it, which is in the temporal lobe. Whenever someone damages that, you get visual agnosia. Or, oops, sorry, you don't get that. You get prosopagnosia, which means you can't perceive faces, but you can perceive common objects. The opposite of that is visual agnosia, which is where you damage the inferotemporal cortex, and you can't perceive common objects anymore, but you can perceive faces still. It's the only object really that you can perceive. This means that we have a double dissociation in these areas. That means that you can damage one area of your brain and it doesn't affect processing in other behaviors. So for this, you can damage the fusiform face area and all of your other behaviors totally fine. It's just your ability to process faces that is impacted. Or vice versa with inferotemporal cortex. And I got a video, in case this is kind of hard to describe or hard to understand just through me describing it with words. This video will walk you through it, and I feel like does a pretty good job of showing you the day-to-day struggles that exist for people that have prosopagnosia. Alright, so you may remember that we've also talked about, and I already named the area, the fusiform face area, but it should have been a little bit familiar to you because we've talked about it a couple of chapters already. In chapter 2, we were talking about neuroimaging examples, and then in chapter 3, when we were talking about how top-down regulation means that you can look at this face and you can either focus on the face or focus on the house, and depending on which of those you're focusing on, it changes the activation of your brain, right? If you're focusing on the face, you're going to have more activation in the fusiform face area. The fusiform face area is one of the most common or most reliable findings in neuroimaging. Neuroimaging is very, very difficult. As we talked about in chapter 2, there are a lot of inferences you have to make whenever you're analyzing this stuff. And analyzing stuff, I already told you, it's really, really hard to analyze neuroimaging data because the data is so noisy and there's so much of it. But even so, if you're interested in face perception, you got your work is a little bit easier because you're almost always going to find this area active whenever you have somebody looking at a face. It's one of the most commonly easy to find things in neuroimaging. And and I say that because, like, for example, the amygdala, which is responsible for emotional processing and stuff like that. We talked about that in chapter 2. I was in a neuroimaging study where we were trying to find differences in amygdala activation between soldiers that had PTSD and soldiers that did not have PTSD. But it was so hard to establish finding the amygdala and also getting it to be consistently active that it was just really hard to do. The face is much easier than that. You show somebody a face and they're almost always going to have that area of the brain active, even whenever they are not necessarily consciously aware of that. So the $1 million question that was tossed around whenever we were first realizing that the fusiform face area helped us process faces was this: are we biologically prepared to detect faces? To phrase that a little bit differently, does this mean that we have an area of our brain that is dedicated to processing faces because we know that that will give us an evolutionary advantage? Why would that give us an evolutionary advantage? Because we would be able to see a face and know if it is a friend or a foe, or know if it's a family member or an enemy, or know if it is another human or if it is a gazelle or a dog or a wolf or whatever. There's lots of, and obviously that all seems like easy things to do, but this actually needs to be in place for babies and children too, because we need babies to be able to survive and know, hey, this is your mother or father, right? Because they can, babies can recognize faces early on, like that. And we'll talk more about, actually we won't talk about that too much. If you want to learn more about that, you got to take sensation perception, or, child development. So is it the case that we have a part of our brain that naturally detects faces? That's the question here. So, There was a series of articles that came out in the turn of the century from Isabelle Gauthier, who was working, I think, at the Chicago Urbana-Champaign, that university, a very prestigious university in terms of neuroimaging research. They do a lot of really great stuff there, a lot of cutting-edge stuff, computational, cognitive neuroscience stuff there. So her lab was really interested in this idea of the fusiform face area, and she believed that there were other stimuli that could elicit the fusiform face area to be active. And what she found is that it was almost completely just things that we had other experiences with. So basically she found that whenever you have car experts look at cars, that the fusiform face area would be active whenever people are looking at cars. What? That's bizarre. Why would that happen, right? That's so strange. But in that same study, she found that people who were bird experts, when they would come in and they looked at birds, they'd have the fusiform face area also active. So whenever they were looking at birds, it was also active. But the interesting thing is, if you had a bird expert look at a car, no activation in the fusiform face area. And if you had a car expert look at a bird, no activation in the fusiform face area. And so this just kind of shows this. I'm not going to go into the data too much here, but you can kind of see that for bird experts, they have a lot of activation, a little bit of activation, when they're looking at faces. So we've got the fusiform face area active in both of these. Now when the car experts are looking at cars, we see that same area is active, but not for the bird experts. But for the bird experts, when they're looking at birds, we get that area active, but not for the car experts. All right. So the question here that lingered, that was, how can you, so thinking back to research methods 1, right? To talk about an experiment, you need to be able to have some kind of random assignment. You need to be able to manipulate groups. You need to be able to take random people and put them in either group. That's not really possible in this study because you can't randomly assign someone to be a car expert. One of my oldest friends, somebody that I knew when I was like 2 months old and grew up with him all through high school. His name's John Hamner. John, if you're watching, I haven't talked to you in forever, man. I should reach out to you, man. We hadn't talked in over a decade. We had so much in common. In high school, we drifted apart because we had different interests. I'm talking about you now because I haven't forgotten about you. That you were a car guy. You loved cars and you loved trucks and you knew everything about them. And you did that because over years and years and years, you were interested in it, you were reading about them, you had magazines about it, you worked on cars, and that's now what you do for a living. You're a mechanic. So I couldn't randomly be a car expert because it takes years to become a car expert. Just like it would take years to become a bird expert, right? There are people who are they call them birders. They go around and kind of collect observations of birds. It's a hobby that people have. But my point is, I couldn't take a random person and say, I want you to be a car expert, or I want you to be a bird expert, because that means that for the next 5 years of their lives or whatever, they have to specialize in this one topic, and that's just not realistic. So that brings us to this study right here, where, again, Gauthier and colleagues realized this problem with the other experiment, so they were like, we need to be able to randomly assign people to a group so that we can establish cause and effect. So they wanted to have a genuine experiment. So instead of finding people who were already experts, they decided to come up with an experiment where they could train people to become experts in a field. How exciting, right? But what field would they be experts in? They created a totally new field, totally new stimulus that no one had ever seen before, and they called them greebles. Greebles look like this. They look like these weird they look kind of like goblins, I guess. Or to me they do. But they don't really look like anything, right? Like, besides that, they don't look like anything else I've seen. They don't like cups, they don't like cars, they don't like the birds. They are completely new because this lab, Gauthier and colleagues, they created these stimuli specifically for this purpose, to test people on a stimulus they had never seen before and make them experts in it. So the way they did this task is that they would basically come in and have people do a memory task. So that's what we're going to do here. So for the common objects group, or condition I should say, they're going to do a memory task where the question is this, have you seen this object before in this task? So the first one, you haven't seen it, is the first one, so you would say no. Now I'm giving you a visual mask. This is called a mask. It's mentioned in the textbook. A mask is going to be a stimulus like this that essentially kind of resets your perceptual system. What do I mean by that? If I show you this penguin and you close your eyes, you can still see the penguin, right? Or if you were to look at this and then if I flashed this so that it was a white background, you'd have the after image of the penguin there still, right? So, and actually if I can do this. So you're looking at the penguin, you're staring at it, and then when I bring it up to this one, you can still see the outline of a penguin in your after image. So to prevent that, we have a visual mask. This visual mask is meant to disrupt that, so that way you don't have any after images. So when I ask you, have you seen this, you would say yes, and you saying yes is because you recognize that you remembered it and not because you had some kind of after image allowing you to identify it. All right, have you seen this yet? No. No. Yes. No. No. Yes. Yes. No. Yes. No. Okay, pretty easy, right? There's a lot of reasons why this particular task was kind of easy. We'll talk about that in Chapter 9 when we talk about concepts and categories. Now let's do the same thing for faces. So we're going to be doing the same kind of task, but we're going to be doing it with faces. So here we go. You haven't seen this face yet, so you'd say no. All right, what about this one? You would say no. I'm going to, well, hold on. I actually don't remember. Okay, that is a different person. Whoops. So I'm not super good with faces. Long story. But if you take me in the classroom, you know that I will recognize everybody's faces and match it with their names, because I take that very seriously as a teacher. I want to get to know everybody's names and faces, although it doesn't come naturally to me. So I would have gotten this wrong. I would have said yes, even though this is the first time we've seen this individual. All right, what about this person? Yes, we did. No, not yet. Yes. Yes. No. Yes. No. Yes. Yes. Okay, cool. All right, so we're going to do the same thing one more time, but now we're going to do it with greebles. Here's the thing. This one is going to be the hardest condition of all. And the reason why is because for the other two conditions, you have experience with recognizing common objects because you've been doing it all your life. The same thing is true for faces. You haven't really looked at greebles before, so this is going to be kind of hard. Here we go. All right, you haven't seen this one before, so you're going to say no. I'm going to do this. I'm going to do my best shot on this. I'm not sure if I'm going to be accurate, so if I'm not accurate, you can send me an email and tell me, and then I'll just delete it without reading it. No, I'm kidding. I'll read it, and it'll be very funny if you go through and check me on this. All right, so we got this guy. I'm going to take it away. Yes, we did see this one. No. No. Yes. No. No. Yes, I think. Ooh, I think so. Yes. 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 I don't know. I don't know. That one was so much harder than the other ones. So basically, the researchers in the study, they were giving people common objects to recognize, faces to recognize, and these greebles to recognize. And then, they would essentially teach this group what the names were for these greebles. So this greeble right here would be called a Doral or an Enoch. Basically, they randomized it so that half of the group got this word and the other half got this word. Why do they do that? Just to make sure that there wasn't something specific about the word doral, which made this easy to remember, so they gave each of them 2 separate names and randomized it so that way they could compare within these groups. So you can see this gets harder now. I'll let you try. So have you seen, in this task, have you seen this one? You haven't seen it yet. I'm going to take it away. What about now? What about now? What about now? And so you can see it's even harder whenever you have to, whenever these things are manipulated, whenever they are turned or rotated or enlarged or shrunken or tilted, it gets much harder. So in this task, they had participants trained up on these greebles to make them good at it. And they trained them up for, I want to say, 5 to 7 days. So not really all that long. But before training, whenever her participants came in and they gave them this task, basically looking at these greebles resulted in no fusiform face area activation. But after training, after people had some time where they got to know these stimuli, they got pretty good at it. And not only did their accuracy go up, but you can see that it also changed the activation in their brain. So we're looking at for novices here. So this would be like the very, this is the very first day. So we'll say day 1. And we'll say day 5 over here. So in day 1, when people are looking at faces, the fusiform face area is active. Cool. But we would have expected that. So that's not, so who really cares all that much? But when we talk about greebles, nothing. Doesn't matter. You don't see any greeble activation here, or fusiform face area activation when people are looking at greebles. But after 5 days of practice, we see that yes, people, their fusiform face area is active whenever they see faces, but now, after just a few days of practice, you can already see some areas of activation in the fusiform face area. Which means that, and this is kind of the big take-home message, is that the fusiform face area does detect faces, but really what it does is it detects and helps us discriminate stimuli that we are familiar with. If you are a bird expert, it's going to help you discriminate birds. If you are a car expert, it's going to help you discriminate cars. If you are a painting expert, if you're an art historian, it's going to help you recognize the difference between Monet and Van Gogh or something like that. All right. So the last thing that I want to mention is holistic processing. In part because this is going to be directly relevant, is what I was trying to say. It'll be directly relevant to our first research quiz, which is about face processing. So basically, holistic processing is this idea that when we look at a face, what we recognize in it isn't the specific eyebrow or the specific nose shape or the specific bottom lip of any given face. That's not how we recognize it. Instead, we recognize the combination of these features. So we recognize the distance the eyes are from one another. We recognize the distance from the eyes to the bottom of the mouth. And if that looks familiar, people will call that usually like a T formation. And when we talk about like face recognition, those of you that are in criminal justice, I know a lot of you are either criminal justice minors or criminal justice double majors, that when we talk about technology that helps us recognize faces, this is what they're responding to, is the T formation. They're not, detecting moles on our faces or wrinkles on our faces. They are detecting the way that our eyes and our mouth and our nose are combined and where they are in relationship to one another. That is called holistic processing, which means that I could go in and I could change. This is Harrison Ford, famous actor. I could change his eye color and you still recognize him. Or I could take out his earring and you still recognize him. Or I could change his teeth and you would still recognize him, right? But if I did something like moved his eyes further apart, then he'd be harder to recognize as a human. Or not as a human, but as Harrison Ford, because I'm disrupting the relationship that those features have in common. That's going to be important when we talk about research quiz number 1, where we talk about inverted faces. So don't forget the inversion effect that I talked about at the very beginning of our face discussion here, because that plays a big part in that article. All right, so thank you so much for tuning in. That is it. If you have any questions about the first 4 chapters, feel free to email me. I'd be very happy to talk about it with you. If anything is confusing, because we have our exam, and our first exam is going to cover these first 4 chapters. So that's all that I have for you. So thank you so much for watching and for hanging on till the end. I already showed you a picture of, or I already showed you my dog. He's still, he's under the desk here. So yeah, I don't know why I'm stalling. I don't want to let you go. I keep waiting here, like you might ask me a question. So maybe I hear you typing up the email right now, and maybe your question is, Professor Daniel, have you no shame, or something like that. I don't know. All right, see you later. Bye.